introduction of everyone. 1 minute. One minute. I'll just share the screen. Can you see my screen?
Yeah, we can see.
Okay, great. So, as you all know, today we are discussing about sickle cell disease because today is the sickle cell day, and this day is being celebrated to increase the awareness about the sickle cell disease. So today we have a panelist with us, Dr. Dipty Jain. She's actually a leading expert in sickle cell disease in India, and she has done a tremendous work in the patient population suffering from sickle cell disease. And we are very, very glad to have Dr. Dipty Jain with us. The next expert is Dr. Akshay Sharma. Dr. Akshay Sharma is a attending consultant at St. Joe's Children's Hospital, and he is having a special interest in sickle cell disease and is working on stem cell transplant, making them very safe for these patients, and also working on gene therapy in sickle cell disease. Welcome, Dr. Akshay Sharma, and thank you so much for joining us at such a short notice. Then another expert we are having today is Dr. Atish Bakane. Dr. Atish Bakane has worked with Dr. Gaurav Kharia also. Dr. Gaurav Kharia, we know that he has done a lot of sickle cell disease transplants in India. And Dr. Atish Bakane himself is now working in an institute in Nagpur and is recently done few transplants in sickle cell disease patients. Now, other discussing with us is Mr. Gautam Dongre. He is a very well-known person in the sickle cell disease community, and we are very glad to have him. Thank you for joining Mr. Gautam Dongre. He is the Secretary of National Alliance of Sickle Cell Disease Organizations. And nonetheless, Dr. Pankaj Parekh is there with us. He is our mentor and a senior consultant in Paediatric department, and he always support us and encourage us to take new things and start new initiatives. I'm very glad that he took some time from his busy schedule and has joined us for this discussion. So with this, we'll start our meeting. This is just a overview about the sickle cell disease for the people who have joined the webinar and to understand what exactly is sickle cell disease. So as we know that the sickle cell disease have a normal RBCs which are circular in shape, but because of the deoxygenation, the sickle the RBCs who have a defect in their gene become sickle.
Shweta, we can't hear you.
Oh. Can you hear me, sir, now?
Now we can hear.
Okay. I think there's some issue with this thing. So, so I'll just take from this sickle cell disease happens because of the abnormality in the gene, in the hemoglobin gene, and because of that mutation, there is the deoxygenated RBCs, they acquire a shape which is known as a sickle shape. Now, because of the sickling of these cells, we know that these cells lead to hemolysis, lead to vaso-occlusion, lead to vesiculopathy. And because of these hemolysis, we have jaundice, we have ulcers, we have pulmonary hypertension, we have strokes. And because of the vaso-occlusion, it leads to the hypoxia, there is an acute chest syndrome, it leads to end organ damage, nephropathy, and vasculopathy leads to hypercoagulopathy and inflammation. So sickle cell disease is actually quite different from the beta thalassemia because sickle cell disease actually affects each and every organ of the body because of the chronic hypoxia also and because of the chronic inflammation also. It is not just anemia, it is actually a conundrum of a whole disease. So now I will not go into the detail of this. We all know that it is a monogenic disease which is happening because of the substitution of a single amino acid. So there is a misspelling. Like if we are having a word like call.
Shweta again
call.
muted.
Okay. So we have a word like call. Can you hear me, sir, now?
Now very well.
Yeah. So we have a word like call, and what happens with that word, if there is a misspelling is there, instead of call we are writing calm, calm. So there is only a single substitution of from L to M, but the whole meaning of the word changes. The similar thing is happening over here, where the single amino acid substitution is happening. Now we know that the inheritance pattern of the sickle cell disease is also quite different. And in this, we have like if the mother and father having a different combination, whether the father is straight or mother is normal, or whether they are both are having sickle cell trait or both are having sickle cell disease, accordingly their progeny will have a effect. There will be a proportion of the patients children who will be normal. Some of them will be carrying the trait, and some of them will be carrying the disease, depending upon what the mother and father are carrying, what kind of genes. Now in India, there is a high prevalence. You can see there is a belt of sickle cell disease. It is present in areas of Maharashtra, that is Vidarbha region. Then in Madhya Pradesh, almost out of 45 districts, 27 districts are affected by sickle cell disease. Then there are areas of Chhattisgarh, Odisha, areas of Rajasthan and Gujarat. So this belt is a sickle cell belt, and it almost the prevalence of the sickle cell disease is almost uh, 10 to 40% in this population. So very high prevalence of sickle cell disease is there. So now we have certain drugs for the sickle cell. So we can treat them with hydroxyurea, and there are other drugs which are in trial, and some of the drugs are available. But in our Indian population, hydroxyurea is the main modality of treatment and the pain crisis by the pain medicines we use. Now, my question to the panelist is management of the disease. First of all, it is very important to know or understand what is the profile of the disease in India and whether it is different from the African population, where it is more severe, and many of those patients in African population go for a stem cell transplant upfront. So now what is happening in India? So my first question is to Dr. Dipty Jain. Ma'am, what is the profile of patients in India which you have seen in your clinical practice with symptoms like VOC, ulcers, pulmonary hypertension? What are the complications you see in these patients?
Thank you, Dr. Shweta. First of all, I would like to say that the haplotype in India is different from African. We have Arab Indian haplotype, which is characterized by very high hemoglobin F. The mean hemoglobin F percentage in our country is around 14 to 22. Between 14, that is the range, which is very different from African. So that is the basic difference. And hence, there was a general belief that Indian sickle cell disease are milder, but that is not so. In spite of having high hemoglobin F, the sickle cell disease in India is very heterogeneous. I can't say it is very severe everywhere or mild. It is different in different places. So it is in India, central India, I know it is very severe. And the commonest phenotype, I would say, is triad, for which we get admissions, are pain, anemia, and infection. These three are the commonest phenotype. However, you do get recurrent jaundice regarding leg ulcer and priapism. This is not seen as commonly as seen in African population, even pulmonary hypertension. It is not as common as we see in African population. However, renal involvement, renal failure is seen quite commonly in our patients. Shweta?
Okay.
Done.
Yeah. So ma'am, so there are certain symptoms which are actually there more prevalent or quite common in Indian population. But as you said that ulcers and pulmonary hypertension, we don't see that commonly. So this is the study which was actually published by Dr. Kanjaksha Ghosh, and there was a perspective in which they have tried to find out the difference between the African disease versus Indian disease, and they have found that the pneumococcal septicemia complications are considered as high in African population, but not so much in, or it is actually unknown in Indian population. Similarly, stroke incidence is quite high in Indian disease. We don't know what is the prevalence of this, but most importantly is that this study has been done in 2016, and still we don't have much clear data about the profile of our patients. So maybe now we know that ICMR is closely looking into it, and even the Indian government is closely looking into the sickle cell population and the profile of these patients, and we'll have more data about this modality. So my only Mr. Gautam, since you have seen sickle cell patients in your family, so what is your experience, and what do you think about the facilities which are available in India?
Thank you, Dr. Shweta, for giving me the opportunity. जी हाँ, मेरे घर में अभी 2 बच्चे हैं। मेरा बेटा 18 साल का है और बेटी 13 साल की, दोनों सिकल सेल वॉरियर है। जब वो उसका जन्म हुआ था और तकलीफ में था, तब की कंडीशंस कुछ और थी। तब ज़्यादा अवेयरनेस नहीं था, कम प्रमाण में था, मेडिसिन के बारे में उतनी जानकारियाँ नहीं थी। लेकिन धीरे धीरे जैसे समय गुजरता गया और बाद में हम लोग नागपुर आए और ट्रीटमेंट हमारा जीएमसी डॉक्टर दिप्ती जैन मैडम के यहाँ से ही शुरू किए। उसके बाद समझ में आया कि हाँ, हम लोग अब जो सिकल सेल पेशेंट है उनके लिए कुछ ट्रीटमेंट है, कुछ प्रोटोकॉल है, और वो इनके सहारे एक अच्छा जीवन जी सकते हैं। सिटीज में आने के बाद ये सारी चीजें पता चली और फिर वैसे हमने अपने बच्चों को रखना शुरू किया। और अब वो प्रॉपर ट्रीटमेंट में है, प्रोटोकॉल में है, हाइड्रोक्सी यूरिया रेगुलर ले रहे हैं। और अभी ठीक है, उनका जो लाइफ है वो नियर टू नॉर्मल मैं बोल सकता हूँ। ये हो गया मेरा फैमिलियर, मेरा एज ए पेरेंट, बट एज ए ऑर्गेनाइजेशन, एज ए सिकल सेल रिप्रेजेंटेटिव, अगर मैं इस चीज को बोलूं तो सिटीज में तो ठीक है, लेकिन जो रिमोट में हमारे जो पेशेंट्स है, जो सबसे ज्यादा है, उन तक जहां तक अवेयरनेस नहीं पहुंचा है, जहां मेडिसिन नहीं पहुंची है, उनकी हालत बहुत खराब है। वहाँ असमय मृत्यु भी हो जाती है। वहाँ क्राइसिस हर 1.5-2 महीने 3 महीने एज पर कंडीशंस आते हैं। कई जगह तो ऐसे है जहाँ डायग्नोस नहीं हो पाए हैं, डायग्नोस नहीं होने से उनका ट्रीटमेंट शुरू नहीं हुआ है, और उस वजह से वो रेगुलर पेन क्राइसिस में है, और सडन डेथ के भी केसेस काफी हो जा रहे हैं। तो टोटल अगर इंडिया का सिनारियो देखेंगे तो भारत सरकार काफी मेहनत कर रही है पिछले कुछ वर्षों से सिकल सेल डिसीज पर, और कुछ हद तक उन्होंने बहुत सारा अवेयरनेस और बहुत काफी जगहों पर हाइड्रोक्सी यूरिया जैसे ट्रीटमेंट, ब्लड फ्री, और डॉक्टर्स भी काफी जानने लगे हैं। लेकिन अभी भी काफी ऐसा एरिया है जिन्हें कवर करना बाकी है, और वो जितने जल्दी कवर हो उतना अच्छा, क्योंकि हमारे सिकल सेल पेशेंट्स के पास समय कम होता है, क्योंकि ईच एंड एवरी क्राइसिस, पेन क्राइसिस मैटर्स, और ऑर्गन डैमेजेस जो हो रहे हैं उस वजह से उनकी डेथ जल्दी हो जाती है। तो सिनारियो टोटल ऐसा है कि जो जहां पर सिकल सेल डिसीज का ट्रीटमेंट प्रोटोकॉल है और अवेयरनेस है, वो एरिया ठीक है, लेकिन बट रिमोट एरिया में तकलीफ अभी भी बनी है। थैंक यू।
Thank you, Mr. Gautam, for sharing your experience and the issues which are actually ground realities. and we talk about sickle cell disease. We are talking about the various things nowadays, but definitely we have to reach a single person, every single person in India who is suffering from sickle cell disease. Then only we can say that we are making a progress towards the cure of this disease. So my next question is to Dr. Pankaj Parekh, sir. Sir, what do you think? What we can so what is your experience, sir? Once a patient comes to you, what are the issues you face when a patient comes to you with these problems which are can you hear me now? Can you hear me now?
Sorry, but couldn't hear the question only.
अच्छा सर अभी आप सुन सकते हैं मुझे?
हाँ,
Can
now
you hear
clear.
me now?
Now clear.
Sir, so what do you think, like what more we can do for the sickle cell patients in your experience when they come to you? What are the lacuna you feel in their care as a pediatrician?
Shweta, all these years, over a period of 45 years I'm in a practice, I remember my early days of practice where we didn't have any qualified pediatric hematologist. There were general hematologists, and there was a limited facility to investigate. I'm sure there was a limited understanding of the disease. And we have lost quite a few children in acute bacteremia, plastic crisis, clinic sequestration. And before we could realize, the children used to die. So it is very, very important, I think, that we diagnose sickle cell early, so we are ready for these challenges. Now, there's one important question which I always have been having. When the newborn is born, we have a screening test for congenital adrenal hypertrophy, phenylketonuria, and so many, 5, 6 of them, we do that, right? Why we don't do testing for sickle cell at birth? Isn't it very important to do a sickling or a sickle cell diagnosis at birth? Is it possible? And what are the ways we do, Shweta?
So what are the
No, the new screening, can we include sickle cell disease?
Yeah, I think that this is a very
Shweta,
important
can I
question.
answer this question?
Yes, ma'am.
Can I
Yes,
answer
ma'am.
this? I just
Ma'am,
want
I
to
was
tell him that, yes, actually, we already have now a newborn program, Government of India, which is going to be implemented soon. And you are very right. We must have all newborns screened for sickle cell disease, whose rate is much higher than what we are screening for. You are right, sir. So that the only thing is the test done for newborn is different. We cannot do with solubility and slide test. We have to do HPLC.
Correct.
And nowadays we have point of test, but to confirm, we'll have to do HPLC and put the children on prophylaxis, penicillin, folic acid, and give them special vaccines. I think, and do anticipatory guidance and education, which is very important. Sequestration crisis you can prevent. So you are right.
So now we have discussed about the diagnostic facilities, and we have discussed about the primary treatment facilities which are available. So we are going to discuss about uh, uh, regarding the frequency of the admissions in these patients. Dr. Dipty Jain?
Yes, can you please repeat the question?
Yeah, I just wanted to understand what is the frequency of admissions of such patients in your clinical practice. Like if one patient is diagnosed with sickle cell, how frequently they require an admission in a year?
Okay. It differs from patient. I told you they are very heterogeneous. Even in the same family, if there are twins which are SS, both one patient will be admitted repeatedly and the other will never be admitted. So it depends. So I would say 20% or 15 to 20% of the patients are very severe one who are admitted, I would say, at least 7 to 8 times in a year.
Okay. So now
For
the
different
question is,
things. Does that
yeah.
answer
So
your question? Yeah.
yes, ma'am.
Yeah.
So basically, I wanted to understand what is the
Hmm.
severity and the quality of life in India patients, because mostly the general impression is that many of the sickle cell patients, they have good quality of life, they are less severe, and they do well on hydroxyurea, and therefore they don't require any curative therapy. So we need to understand exactly which population or
No.
of Indian patients require what kind of a treatment and care, so that we can direct and develop a proper standard of care facilities for these patients.
Okay. Actually, the patients, I would say 7 to 8% of them have stroke
Mhm.
who are not diagnosed from birth. They have
Okay.
stroke. And the worst part about
Mhm. Ma'am, your voice is gone. Sorry.
stroke is must under curative therapy. It's a must. And there
Yeah.
are 5% of my patients who have chronic pain. They're always in pain. This is another group which I would say. So
Okay. So
these
this is
are the
very
patients which I'll go for.
So this is very interesting, because in the as Dr. Kanjaksha Ghosh paper says, it is unknown, the stroke incidence is unknown, and it is almost 10% in African population. But what you are saying is that almost it is 7 to 8% in Indian population also
Yes,
in your clinical experience. So which is
yes,
quite high. Yeah.
yes, yes,
Yeah. So
yes.
now
It is
coming
high.
to,
Yes, because
yeah,
we miss them. We miss them. बहुत बार तो
yeah.
stroke आया कि तुम sickling
Mhm.
करो तो तुमको समझता है कि it is sickler.
Correct. Yeah. So now, when we have understood about the patient's presentation, their clinical severity, and the complications associated with sickle cell disease, and definitely these patients, they require more than hydroxyurea, and many of these patients are actually eligible for the stem cell transplant and other forms of curative therapy. And at present, we have stem cell transplant as the best curative option which is available in our country. Now we can do stem cell transplant by using a sibling or a relative who is completely matched, or we can have a match unrelated donor, or we can have a mismatch marrow donor, that is a haploidentical related donor is there. So my next question is to Dr. Akshay Sharma. Dr. Akshay Sharma, what is your experience in different types of transplant in sickle cell disease, and how you select your donor?
Yeah, thank you, Dr. Bansal. So I'm going to answer this question sort of related to what Dr. Jain just said. I think you have to balance the risk of a stem cell transplant to the risk of the underlying disease. So, like you are showing on the slide over here, if the patient has milder disease, then you shouldn't take a big risk from transplant. For example, if the patient is taking hydroxyurea and they're doing fine, and they have maybe one pain crisis per year or less, and they have no evidence of any organ damage, then I don't think that those patients need to be considered or recommended for transplantation, because no matter how safe we try to make transplant, it is always associated with the risk of death. And that risk of death, as we are improving the transplant techniques, is going down, but it is not zero. So if a patient is doing very well on supportive care, on hydroxyurea and other medications, I think they should wait. But like you have pointed out over here, if they have definitely had stroke or have elevated transcranial Doppler, which increase the risk of stroke, that is the highest risk of patients. And I think those patients need to be recommended for transplant using the best available donor. Now, if they have a sibling, we should go for the sibling transplant. If they don't have a sibling, then I think even unrelated donor or haploidentical donor transplants can be considered for them. It is patients who don't have stroke but have other problems like pain crises or organ damage, cardiomyopathy, pulmonary hypertension. These are sort of patients in that gray zone where the risk of death can be higher. And you can attenuate that risk of death with supportive care. But what if, like Mr. Dongre was saying, if these patients are living in areas where access to these therapies is not easily available, then what do you do? Then in that situation, the risk is higher. So it is sometimes, feasible and even easier to bring these patients to the bigger cities, let's say Bombay or Delhi or Nagpur, where they can live for 6 months or 9 months. We do the transplant, and then these children can go back to their hometowns. So we can, provide them a potentially curative therapy, uh, because if they continue to live in areas where even basic therapies are not available, then the risk of death from these complications can be very higher. So transiently, if you bring them to one of these bigger centers where transplants are available, you do the transplant safely, you continue them on immunosuppression for a period of time, reduce the risk of GVHD, and then send them back. I think that's feasible. But the whole thing needs to be monitored and considered in the setting of what is the risk to that particular patient. And what is the risk of transplant and what is the experience with transplant. Some centers have a lot of experience with transplant, so they are getting much and much better. The risk of transplant in those centers will be lower. Some centers which don't do many transplants, as experience with transplant is lower, of course the risk will be higher. So I think all these factors need to be considered.
Okay. Yeah. So I would like to ask, thank you, Dr. Akshay. It was very clear. I mean, we really like to understand how you are doing the transplants over there in USA. Now we have Dr. Atish Bakane, who has actually performed transplants here in Delhi as well as in Nagpur. So, Dr. Atish Bakane, can you share your experience with us, like about the transplants in sickle cell patients?
Sorry. Sorry. So, I think, as it very rightly put forth by Dr. Akshay, the transplants for sickle cell disease is not, till today, it is not very much well developed or very much well accepted because of the gray zone in indications of transplant in sickle cell disease. Like thalassemia, we counsel that it is better to go ahead with transplant. But sickle cell disease, I think, is a disease of great. It's an enigmatic disease. It's a great phenotypic variations. Whatever, like we have been saying that whatever is HbF, we have seen severe sickle cell disease with good HbF percentage also, and with less HbF, less than 20, also patients are not symptomatic. So that is why I think transplants in sickle cell disease becomes a tricky zone. So according to me, like patients, 5 to 10% of the patients are definitely in the transplant is indicated in them. Most of the patients on hydroxyurea do well, we all know. But 5 to 10% definitely have problems, one or more problems like severe vaso-occlusive crisis, stroke. And in Indian patients, as Madam has rightly told, 5 to 7% or 8% patients do develop. So our understanding was not like the Indian patients do not develop strokes. No. So these are the indications, and moreover, what Dr. Akshay has pointed out in our Vidarbha, particularly because being from Vidarbha, I know what happens in periphery. Most of the patients, they are not able to come to the city itself. Forget about the taking hydroxyurea regularly, or transplant is far ahead. So if we can counsel them, and if they, the specific patients who are indicated for transplant, by arranging money or arranging funds, if we can transplanting, I think it is a good idea. And yes, regarding the type of transplant, matched sibling, matched unrelated, or haploidentical, I don't think at the present moment what type of transplant we are doing matters too much if we are doing at the appropriate age. because in sickle cell disease, we know as they age, the end organ damage is significant. If some patients, many patients who are not on hydroxyurea, or if they don't take hydroxyurea for some or other reasons, they develop problems related to end organ damage like kidney. So we have, I think, faced these challenges in Apollo, because most of the patients suddenly they start having symptoms at the age of 15, 20, and they come. So many patients have some sort of nephropathy, and then it becomes difficult at that point of time to advocate for transplant. It is a very difficult situation. We know, it has been proven in multiple studies that by the age of 12, the outcomes of sickle cell disease transplants are very good, up to 12 and 13, and rightly so because of these reasons. So, like, if matched sibling donor is not available and patient is definitely a candidate for transplant in accepted in a appropriate center with well-experienced centers in sickle cell disease transplants, we can well go ahead with mud transplant. The outcomes are almost similar. In our own data of Apollo Hospital of 120 transplants, so even haploidentical transplants, the outcomes are similar if you do in appropriate age group, like early age group, less than 10 years, the outcomes are good. Unfortunately, as they age more than 15, 20, even with matched sibling, we face a lot of challenges. So I think this will be my take. So if indicated, we can well go ahead with transplants in sickle cell disease. The additional advantage of sickle cell disease transplant is, like, even if we know that graft failure is a bit higher, but mixed chimerism does the job. So that is a very, most of the patients, those who have uh, reconstituted, self-reconstituted, they also have less symptoms. So 20 to 30% of chimerism also is good enough. So I think, with the in last 5 to 10 years, with advancement and with refinement of protocols, the transplant has become a lot safer. I would not say the safest, because there are some challenges. There is definitely a risk of 4 to 5% death in transplant. So we do not advocate for everyone, but as wherever there is a case, and if we are able to transplant them, should be transplanted. Thank you.
Yes, yes. Thank you, Atish. It was quite. I mean, we are still getting now, like
Voice is breaking, ma'am.
best to be transplanted and which patients still don't require transplant. So thalassemia, the transplants are becoming like standard of care now. We know that every thalassemia patient requires a transplant. But in sickle cell, still we are struggling, like, or we want to figure out which patients will be the best to transplant and which patients can be just treated on the hydroxyurea and other supportive treatment. So Dr. Dipty Jain is also here. So I would like to understand from our Indian experts, do you think that it is important for us to develop another severity score which will be applicable for Indian population so that we can figure out and we can mark those patients who are eligible for the transplant and can be transplanted at the early age as compared to the later age when they have more symptoms? What do you think, Atish?
I think ma'am, Dipty ma'am will be a right person to answer this, because why? For the solely for one reason. I think I have pointed out this disease is absolutely difficult to judge. We have seen patients who do not have symptoms till 12 years, 13 years, and suddenly the last transplant I did was the earlier, the older sibling, they lost in 1 year span. The patient was not diagnosed to be sickle cell disease till 10 years of age, and 11th year she started having VOCs, acute chest syndrome, and so severely that one of the acute chest syndrome, she was not been salvaged. And that is why their younger sibling came to us, who also had a problem with hydroxyurea, very severe intolerance to hydroxyurea, had developed stroke. So it becomes very difficult, I think, like other diseases, when we can pinpoint some risk scores. In this disease, it is really difficult unless there is some stroke or some, these are the very specific pointers. but other things very difficult to explain. In my, I think, short, well, maybe span of 10 years, what I have understood. I think Madam can elaborate on this because she has seen sickle cell disease for long. Dipty Jain, ma'am, please.
Right. Yeah, Dr. Jain, can you tell us? Yeah, ma'am.
Or Gautam Dongre sir also, because he is using lot of phenotypic. So phenotypic appearances, phenotypic correlations we have seen in sickle, but it's very difficult to pinpoint those scores. So I think Gautam Dongre ji also can pitch in here.
डेफिनेटली सर. देखे हैं हमने भी इस तरह के पेशेंट्स की. कुछ पेशेंट्स जो है ड्रॉक्सीयूरिया पर भी रिस्पॉन्स भी कर रहे हैं, और उनका जो एक्शन है वो जस्ट लाइक अ थैलेसीमिक पेशेंट के जैसा है. हर महीने उनको ट्रांसफ्यूजन लग रहा है. लेकिन ऐसे पेशेंट्स कम है, लेकिन है. हर एरिया से एक व्यक्ति ऐसा निकल कर आ रहा है, हर डिस्ट्रिक्ट से एक या हर स्टेट से एक दो ऐसे कंप्लेंट्स आते हैं. बट जिनको ऐसी तकलीफें हैं उनको बोन मैरो ट्रांसप्लांट जरूर करा लेना चाहिए. लेकिन वही तकलीफ है कि थैलेसीमिया पेशेंट को तो ऑल इंडिया के तरफ से 13 लाख रुपीस मिल जाता है, लेकिन अभी भी सरकार सिकल सेल को उस तरह का कोई सुविधा नहीं दे पाई है. तो आज के फोरम इस आज 19 जून जागतिक सिकल सेल दिवस के उपलक्ष्य में अगर ये बात सरकार तक पहुंचे कि जो जिन्हें जरूरत है ऐसे पेशेंट्स की अगर जान बचाना है तो एटलीस्ट उन्हें भी थोड़ा फंड मिले, फिर वो कोल इंडिया की तरफ से हो या और किसी भी संस्था की, उनको भी जरूरी है. Thank you, sir.
Yeah, yeah.
I'll just
So
add
I
one thing
think,
to this. So what Dr.
sorry,
Bakane just mentioned.
Akshay, you were saying something? Yes.
Yeah, no, I was just saying that Dr. Bakane just mentioned, and I think that's quite important. So, we have done a lot of these genotype-phenotype correlation studies here, where we have looked at different genetic predictors of outcome. So there are a number of different genes, polymorphisms in BCL11A, ApoL1, etc. So we know that some of these genotype-phenotype correlations exist, and we can predict disease severity by just doing whole genome sequencing, which is becoming very, very affordable now. But one of the issues that I must point out is none of these studies are performed in the context of populations outside the African genotype. So these studies are either performed in Europe or they are performed in the US, where, as you know, majority of the haplotypes that are seen are of African origin. So the Arab Indian haplotype, which is seen in India, which it may have a completely different genotype phenotype correlation. And one of the things that we know is that Arab Indian phenotype has a much more heterocellular expression of fetal hemoglobin, while with the African genotype, when they have HPFH, the hereditary persistence of fetal hemoglobin, they have a more pancellular expression of fetal hemoglobin. So 10% fetal hemoglobin in an African American is very different from 10% fetal hemoglobin in an Indian patient. And I think that's the critical piece, that we haven't done those studies to look for what are the genetic correlates of disease severity and whether the expression of fetal hemoglobin is pancellular or heterocellular, which determine what the disease severity is going to be like. So I know ICMR is now interested in this and are paying some attention. So the first thing that probably needs to be done is to have a large registry of patients where these studies are performed so that we can define what the parameters, what the genotype phenotype correlations in the Indian population looks like. I think part of the reason we are having these problems is we in India are making all these decisions based on guidelines that are uh, issued in the US and Europe, which are based on a completely different patient population, and that is not applicable at all to patients in India. Some things, okay, hydroxyurea, good, it works, but how it works is unknown, and it may work differently based on what the genotype is. So first of all, I think it's very important that we do these studies in the Indian context. We define what the genotype of Indian patients looks like, what the disease expression in Indian patients looks like, what is the long-term outcome. We know that in the United States and African Americans, the average lifespan is 42 years. And we also have a very good description of what the disease phenotype look like when they develop pulmonary hypertension, when they develop cardiomyopathy. But these data in Indian population is missing or is very limited. As you mentioned, in 2016, that study was published. And now we are, 7 years later, and we still don't have an answer to that. I think this is the first thing that we need to advocate for, and we need to talk to the government or ICMR, that these are the things that we first need to define so that we can identify what would be the indications for transplant,
Exactly,
who are the
exactly.
patients who are going to have the
Yeah.
more severe phenotype, so that then we can put the right resources in the correct place.
Exactly. Akshay, this thing has been brought up very nicely by you. Actually, we don't have data about Indian population. So whatever the indications we are seeing for the stem cell transplant, which is different for the mass sibling, this thing, we don't know how Indian population is behaving, and we are just extrapolating the data from that whatever the population, African population, then we are trying to put it on Indian population. Maybe Indian population is quite different, and they require a different strategy for the treatment, and at least to improve the quality of care, we need to have that kind of a data where we can do the genotype and phenotype correlation. So Mr. Gautam Dongre, मेरी आपसे रिक्वेस्ट है, मे बी वॉट एवर वी आर डूइंग, अंटिल अनलेस हमारे पास एक बेस लाइन डेटा जब तक नहीं होगा, तब तक हम लोग वो क्वालिटी ऑफ केयर जो है वो हम लोग अचीव नहीं कर पाएंगे. और अगर हमें क्वालिटी ऑफ केयर अचीव करनी है तो हमारे को ये स्टडीज करना बहुत जरूरी है, बिकॉज देन वी कैन डायरेक्ट आवर रिसोर्सेज, as Dr. Sharma has highlighted, that we need to direct our resources to the right person, to the right patient in a right way. So Dr. Akshay Sharma, you are doing gene therapy also. So this is my last question. We know that CRISPR gene and gene editing and gene therapy by transduction is being happening. So what do you think, what is the success rate and how it can be brought to India also? How we can bring it to India?
Yeah, so the main advantage of gene therapy is that we don't have to find a donor, that every patient can be their own donor. So that's the main advantage of doing this, because first of all, what that means is that every patient can potentially get gene therapy, and secondly, there are no immunological complications, so you don't have to worry about graft versus host disease or rejection. So we have done quite a few of those at our institution, and of course, as you know, there are a number of studies which are going on in the United States and Europe also. I think the biggest challenge is manufacturing of these cells. They require very specialized equipment and GMP facilities and personnel who are trained in the manufacturing of these cells. I strongly believe that these things can be done in India, and there are centers in Bombay and in Vellore where they are already making CAR T cells. So the equipment and the resources are very similar to manufacturing of CAR T cells. So if they can make CAR T cells, they can make gene therapy also. The process is completely different here. So I think this can be brought to India fairly rapidly. And we just need interested parties. So at St. Jude, we are developing our own gene therapy as well. And our goal is to, as soon as we have proof of concept, so we have done studies in animal models where we show that it is effective, and our clinical trial hopefully will be launching very soon. And so once we have done some proof of concept studies in patients over here that we can do this and it works, we are more than happy to then partner with facilities in India where I know that these resources are available to manufacture these therapies, and then we will want to provide these therapies to people in India, because believe it or not, the majority of patients with sickle cell disease are not in the US. They are in sub-Saharan Africa, India, South America. And if we develop all these therapies, it's no point if the patients in places where they are most prevalent, they can't even access these therapies. The other thing also is the cost barrier, where these companies, pharmaceutical companies, which are making gene therapy, they are selling it for a exorbitant cost. I'm sure, as you know, you might have heard, $2 to $3 million. at that price point, nobody outside the US, even in the US, half the population can't afford it. Even as outside the US, absolutely illogical. Like
Exactly.
nobody will be able to afford it. So, and it is not that it takes that much money to make these therapies. It's just that, out of greed or whatever, these companies are trying to make a profit out of patients. And St. Jude has been a non-profit organization, and our goal is not to commercialize these therapies to just make money. Our goal is to make these therapies accessible to as many people as possible. And so, we definitely hope that we'll find partners and they are able to bring these therapies to people in India. So it can be done. It's challenging, but it can be done.
Mhm. So, what is the eligibility criteria for these patients for gene therapy? Is there any particular age group or any particular indication, or all patients are eligible for it?
So for now, because gene therapy is so new and we do not know what the long-term risks are going to be, all gene therapy studies are starting in adults first.
Mhm.
Uh, and as you mentioned, by the time you reach adulthood, they already have much severe disease. And
Mhm.
so initially, because we don't know what the long-term risks are, we will start these studies in patients who are older than 18. But
Mhm.
as we accumulate more long-term follow-up, more safety data, and ensure that these therapies are safe and effective, then I think we can progressively go down in the age group. And I mean, of course, the long-term goal would be to provide these therapies to younger children before they develop organ damage, so that we can prevent further organ damage from happening. So will progressively go down, but I think in the near future, most of these therapies will be available for older teenagers or young adults.
Okay. And one more question. So, regarding the gene therapy, are you doing it by the lentivirus or you're doing a CRISPR gene?
We do CRISPR-Cas9 gene
Okay.
therapy. So lentiviral gene therapy is also effective.
Mhm.
Bluebird Bio is the company which is doing lentiviral gene therapy. But there are some issues associated with lentiviral gene therapy, which I don't want to go into too much detail over here. But CRISPR-Cas9 is a newer technology, so you have to be a little bit more careful with that. But CRISPR-Cas9, in my opinion, it's a little bit more potent, more targeted approach. And so, I think it's a little bit better than lentiviral gene therapy, though, of course, there has not been any head-to-head comparison. So it's hard to say, but it is my opinion that lentiviral gene therapy is not as targeted or potent. And so we prefer to do CRISPR-Cas9.
Okay. So one more question. So we don't have genotype data about Indian population. So for CRISPR gene editing procedure, will require a genotype of Indian population as well, or it can be applicable to Indian population without that as well?
That's a great question. So, all the investigations that have been done, preclinical investigations, they have been done either on Caucasian patients who have donated their cells or on patients with African ancestry. So I think one of the main things that will have to be done before we start doing these therapies in patients is to actually do preclinical experiments on
Mhm.
samples that have been obtained from the Indian patients to see if there is actually the same amount of fetal hemoglobin induction as we see in the patients that we have treated over here. Because if there are, genetic differences, you actually may not see that much fetal hemoglobin induction, and that will defeat the whole point. So that's one of the reasons why we are looking for partners in India who are able to do these investigations in a laboratory from samples obtained from Indian patients, so that we can actually show that the fetal hemoglobin induction is comparable. And that is going to be the first step to then conduct these studies in India. Having said that, we do have a number of different approaches now. There are approaches which edit the BCL11A erythroid specific enhancer. There are approaches which edit the globin locus. And even within the globin locus, there are multiple targets that we can edit. And so there are multiple different approaches, and I think at least one of them will work in the Indian patients as well. It just remains to be seen which one of these approaches is the most effective one.
Okay, great, great, great. Um, Atish, do you have any question for Dr. Akshay?
Can I ask Shweta, Dr. Sharma,
Yeah, ma'am.
are these
But
all
I
in
think,
research mode?
yeah, sorry.
Yeah,
Yes, sorry.
are this on research mode? Gene
Sorry, say
therapy?
that again.
Uh, are this on research mode? Are we
Yes, so
still
all these are being done
including
in
them for
clinical.
trials, the gene therapy?
Yeah, everything that we have done so far is in clinical trials. As you know, Bluebird Bio and Vertex CRISPR Therapeutics have both applied for their licensing application with the FDA. So we are hopeful that both Bluebird and Vertex will get approvals this year. But everything else is going on clinical trials.
Trial basis. Okay.
Yeah. So, ma'am, I think we were waiting for your views also on this because, as Dr. Akshay has highlighted, that we need a very good, robust data on genotypic and phenotypic correlation so that we can direct our resources for these patients. And as your experience and Dr. Atish experience is there, that
Yes.
many of these patients may not be so symptomatic at particular age, but they may become severe in their complications after a certain age. So it's very, very important, I think, to identify these patients as early as possible so that these patients can be offered better curative therapies. So, ma'am, what is your take on that?
Yes, yes. Actually, this is what we are doing with CSIR, genophenotypic.
Mhm.
And I think what
Okay.
I have is the clinical data. And at
Mhm.
CCMB Hyderabad, they have done
Mhm.
NGS
Okay.
for all these patients. So now
Mhm.
the analysis is remaining, I think, and time will only tell us when we look at the correlate the data, we'll get some um, results. But it
Okay.
is being done. We have already done.
Great,
And we
ma'am.
have huge data, huge clinical data. If you ask me, Dr.
Mhm.
Sharma, I'm ready to share with you. But then correlating with the genotype, I need to take from CCM.
Right. Yeah. Great. Mr. Gautam, do you have any questions?
Yes, yes. Not question, just wanted to share, Dr. Akshay, about the gene therapy. Actually, we heard that in India also, the Indian government has started uh, trial research on the gene therapy by CSIR IGIB. So, have you contacted them to support, or is anything to collaborate with you and them? Is anything?
Yes, I am aware of IGIB in Delhi, and we are willing to offer help to anyone
Yeah.
who is interested and wants to pursue. I am in touch with them.
Great.
Thank you.
Pankaj sir,
I have
last
the
word.
phenotype,
Yeah.
but not the lab data. Dr. Sharma,
Yeah, I heard, and we
how can you
can,
help us? How can you help?
so genotype, I think it's a complicated issue because of privacy laws both in the US and India, you cannot share genetic data so easily. So I think as far as patient samples and genetic data are concerned, we'll have to find labs in India which are capable of doing the sequencing and the analysis. And I think it's good in a way because you want to develop the capacity in the same country. You don't want to send all that data out to a different collaborator and then let them all do their own analysis because
Sure,
they don't have an
sure.
inbuilt
No,
interest in that. So, like you mentioned, I think in Hyderabad they do have the capacity. These things are very readily available everywhere. In fact, a lot of labs that do these sort of sequencing studies in the US are the same labs which have branches in India too. So these can be done in India. We'll just have to find the right labs which do it and then do the analysis within the country to make these genotype phenotype associations. I'm happy to connect you to people who can provide some guidance on how to do this, but I think the work needs to be done in India.
Yeah. Definitely, yes. Yeah, thank you so much. It was very enlightening
Okay. Yeah.
actually to understand all these issues. And I think we have to keep on brainstorming about these issues, and definitely will achieve our goal. I would like to have last words from Dr. Pankaj Parekh, sir. Sir, what is your take on with all the discussion? Sir, you are muted, sir.
It's wonderful because there's something which can be cured, permanently. I'm very sure require a lot of government help, above all a political will to support a particular project, because all in our country, it all depends on a political will, not the finances. If the political will is there, there are enough finances, enough people to support the cause, and the work what Dr. Sharma and Dr. Bakane and Dr. Dipty all are doing are so phenomenal. I'm very sure it's just to be motivated, marketed well, because the way in which we get information about thalassemia major on a media, we don't get such information about sickle cell. I'm very sure majority of the people would not even know about sickle cell.
Yes.
When we were talking in one of the meeting, Shweta, and one of the topics what we're discussing is even anticipatory guidance to the parents. They should learn to even screen in all their own child has got thalassemia to find a sudden increase in splenic size, and to know about penicillin prophylaxis, to know about certain immunizations which are so compulsory and so protective and so important.
Right.
So all these things are very, very important. But I think what is above all important is a cure. And if there is a stem cell, if there is a gene therapy, which can then permanently all right. And I think we need to have a help from government and a strong political will. I think together all of you, I'm very sure is never a red carpet. It's always a difficult job to do anything in India, but I'm sure somebody has to start the work.
Yeah. Thank you so much, sir. And I really like the word that something which can be cured should be given a chance for the cure. Yeah. And we are moving towards it. And my heartfelt thanks to all the panelists for joining and giving their valuable time today for the discussion on this topic. And hopefully we'll make some progress towards it. Thank you so much. Thank you,
Thank you.
Dr.
Thank you.
Gautam. Thank you,
Yeah.
sir. Thank you, Akshay. Thank you, Atish. Thank you,
Thank
Dipty
you.
ma'am. Thank you so
Thank
much.
you for
Thank
inviting.
you.
Thank you, all of you.
Bye,
Thank you.
Shweta.
Bye
Bye, ma'am. Bye.
Bye.
Thank you so much.